So it's now my pleasure to introduce Dr. Rick Peek. He's a real leader in H. pylori, understanding the biology and pathogenesis of H. pylori-associated disease, in addition to his day job as the director of the Division of Gastroenterology at Vanderbilt. And of course, many of us know him as he has been the editor-in-chief of our premier journal, Gastroenterology. So, Dr. Peek.
All right, well, thank you so much. I'd like to thank also Rob and Juha for such an inspirational and aspirational meeting they've created. I'd also like special thanks to Rob for switching the order because, as many of you know, you never want to follow Manuel Amieva in a talk. No pressure. But what I'd like to talk to during my time this afternoon is a little bit about the relationship between H. pylori and the microbiome. We've heard a lot about the pathological changes that occur during long-term H. pylori infection. One of the detrimental frameworks that I think is set up comes with the loss of parietal cells, subsequent hypochlorhydria, and the overgrowth of other bacteria in the stomach. You know, I'm preaching to the choir here because a lot of the foundational studies that have looked at the gastric microbiome have been initiated here at Stanford. And one of the earliest impactful publications was from Elizabeth Bick and David Relman in 2006, in which they used 16S profiling to look at the microbiome population in the stomach of H. pylori infected versus H. pylori uninfected individuals. Many studies have now confirmed that, and what they've found is that a common theme is that the diversity of the gastric microbiome is less in H. pylori infected individuals compared to H. pylori uninfected individuals. There have also been clues from different other studies that have suggested that changes in the gastric microbiome may be important in pathogenesis. This is one study that we've heard a little bit about earlier in the meeting. This is from the Shandong province, in which individuals were treated with antibiotics targeting H. pylori. They then looked at the subsequent risk for developing gastric cancer. And they found that indeed, individuals that received anti-H. pylori eradication had a significantly lower risk of gastric cancer. But buried within this paper was the fact that 15 years later, when the cancer risk was reduced, only half the individuals had had their H. pylori successfully eradicated. So 53% still had H. pylori, but they also demonstrated a significant reduction in gastric cancer risk. Another interesting paper that suggested that the gastric microbiota may be important in pathogenesis came from a computational biology journal. This group developed techniques in which they could detect areas of bacterial DNA integration in human tumor samples. And they called these lateral gene transfers. And they found that the type of cancer that had the second highest number of bacterial DNA insertions was gastric adenocarcinoma. But when they looked at the sequence of those bacterial DNA elements, they found that it was not H. pylori. Pseudomonas actually was the highest bacterial read that they detected. And so I think that this has raised an important question. And what are the roles of non-H. pylori constituents of the gastric microbiota in disease outcomes? So mice are a very good model to study this. Many of the major phyla that are in the mouse stomach are also present in the human stomach, including Bacteroidetes, Proteobacter, bacteria, and firmicutes. And Jim Fox's group has really led a lot of the work trying to understand pathogenesis of the gastric microbiome in disease. And they published a study in gastroenterology in which they used NSGAS mice, and these mice were described by Keith Wilson earlier, and they showed that they could look at the gastric microbiome in uninfected NSGAS mice. They could then infect them with H. pylori, and there was a profound change in that gastric microbiota. There was a blooming of the firmicutes. There was a contraction of the Bacteroidetes. But importantly, did this have anything to do with disease? And at least in this study, it suggested that it did. So to really assess this, they then used germ-free mice. So they took germ-free mice shown in green, and then they monocolonized them with H. pylori. And what their end result was looking at the incidence of gastric neoplasia. And they found that 80% of the germ-free animals that were colonized only with H. pylori had no evidence of neoplasia 7 months after infection. And the ones that did all had low-grade lesions. Comparatively, conventionally raised mice, shown in yellow, that were then infected with H. pylori, 80% of those animals developed high-grade neoplasia. We've also taken advantage of animal models to ask other questions, and specifically, can strain-specific factors of H. pylori alter the structure and composition of the gastric microbiota? And for these studies, we used a different animal model. We used gerbils, and we infected them with H. pylori, and indeed, we saw that there were changes in the gastric microbiome. But then we took this one step further, and we repeated these infections, but used a CagA isogenic H. pylori mutant. And when we looked at community structure that is defined by beta diversity, we saw a separation of the population. So in red are the gerbils that have been infected, the wild-type CAG-positive pathogenic strain of H. pylori. In white are uninfected animals, and in blue are animals that have been infected with a CagA isogenic mutant. And you start to see separations of these populations, indicating that the community structure in these animals is different. We also looked at the phyla in these animals, and we found changes that were really induced by the wild-type CAG-positive strain. We saw a reduction in the composition of the Bacteroides populations in the stomach in CAG-positive infected animals. We saw an increase in the phyla proteobacteria. So it seems like these animal models can also allow you to determine whether a single bacterial virulence factor within H. pylori can alter the surrounding microbial community. So this is all well and good. It's easy to manipulate mice. Is gastric microbial dysbiosis clinically significant? Well, several studies have looked at this in cross-sectional trials. So this is a study that looked at the diversity of microbiome populations in the stomach in individuals that just had chronic gastritis alone, shown in green, or had gastric cancer, shown in red. And they showed that there was a significant reduction in the diversity of the microbiome populations in individuals that had gastric cancer compared to those who had chronic gastritis. And even though there was a reduction in diversity, there was an overrepresentation of bacteria that are normally present in the intestine: Rhodococcus, Citrobacter, Clostridia. In the same study, they created a microbial dysbiosis index in the stomach, and as you would expect, the individuals that had gastric cancer had a higher level of gastric microbial dysbiosis. Other human studies have validated this. Again, these are cross-sectional, but this study from China looked at the diversity in the gastric microbiota in individuals that just had superficial gastritis, but then who progressed along the carcinogenic cascade towards intestinal-type gastric cancer. And as you can see, as you move from gastritis to atrophic gastritis to intestinal metaplasia and to gastric cancer, the diversity incrementally is reduced at each step. But in this study, I thought that they did a particularly clever analysis. They were able to look at the strengths of relationships among different bacteria in these stomachs. And so in green are the strengths of the relationships for co-occurring bacteria, and in red are co-excluding. So if you look at these relationships in individuals that just had superficial gastritis, there are a few co-occurring relationships between some of the phyla. But look what happens when you start to look at the relationship in individuals with gastric cancer. It's almost like there's an intentional construction or cultivation of the microbiota in these individuals that potentially could lead to pathogenic outcomes. So one question that's always come up is, well, is this really just due to H. pylori, or can anything that induces hypochlorhydria in the stomach lead to the same changes? And there was a study that directly addressed this. This is from Mark Pritchard, and they looked at the gastric microbiota composition in uninfected patients, controls, patients that were treated with long-term proton pump inhibitors, individuals with H. pylori-induced atrophy, and individuals with autoimmune-induced atrophy. And if you compare the first two groups, they look very similar. The marked difference in this comparison is individuals that have H. pylori-induced atrophy. It's not just suppression of acid alone that leads to these changes. So these studies, I think, have been important in setting the stage for studies going forward, but it's really, I think, critical now to move from association to function. And there was a wonderful study that was published last year in Gut by Kwan and colleagues. And they took biopsies or gastric aspirates from human patients that had either superficial gastritis, intestinal metaplasia, or gastric cancer. They then did gastric microbial transplants of those into germ-free mice. And importantly, H. pylori from the donors, the human donors, did not colonize the mice, but other bacterial species did. And they found that when they followed these mice over a long period of time, they started to see pre-malignant lesions. Specifically, if you look at the presence of dysplasia and metaplasia, in this heat map, presence of these lesions is indicated by green, absence by yellow. The majority of these pre-malignant lesions occurred in mice that received gastric transplants from patients with intestinal metaplasia or gastric cancer, not superficial gastritis alone. When they started to look at the individual species that were overrepresented in the dysplastic lesions, they found some interesting results. So this is the relative abundance of different bacteria in mice with dysplasia in blue or without dysplasia in orange. And for example, Lactospiraceae was overabundant in mice that had dysplasia. But the inverse also was present. Bacteroidetes and Brevetomonas were less common in mice that had dysplasia. So again, this is informative data that I think can set up future studies, but what about trying to do very mechanistic studies to look at individual species and how and whether they are important in pathogenesis? So, we had a wonderful opportunity to collaborate with Jim Fox's group, and for this study we used the populations that were really created by Dr. Correa but have been sustained by the leadership of Keith Wilson and Blanca Piazuela in Colombia. And Colombia is a wonderful human experimental area to try and discern factors that are important in gastric cancer. Almost all individuals in Colombia are infected with H. pylori. But there's a marked disparity in the gastric cancer risk. As noted by Doug Morgan yesterday, in the mountains, there are very high risks for gastric cancer, 150 over 100,000 population. On the coast, it's 25-fold less. So this offers huge opportunities to try and understand differences in H. pylori strains, diets, human genetic backgrounds, and also whether differences in the gastric microbiota may contribute to these results. So with Jim Fox, we his lab generated some preliminary data using whole metagenome sequencing in 20 patients, 10 from the high-risk region, 10 from the low-risk region. And metagenomic sequencing allows strain-level resolution. It also allows you to ascertain differences in not only bacteria, but other taxa, such as viruses or archaea, other kingdoms. And so when we looked at the community structure of these patients, and in this graph, results from the high-risk area are shown in pink, from the low-risk area are shown in blue, we saw a separation in the beta diversity, or the community structure. And the separation was only present when we looked at bacterial populations, not viruses, not archaea. And in fact, of these 20 samples, 35% had mutually exclusive bacterial populations, and these are indicated by the black arrows. So it looks like, at least at this rudimentary level, we can separate bacterial populations based on town of origin, risk of gastric cancer in this population. Another advantage of whole metagenome sequencing is that you can use programs like gene ontology to really try and predict function. And so using gene ontology program, we were able to show that there were functional pathways that appeared to be overrepresented in the samples that came from H. pylori-infected versus H. pylori uninfected individuals in this 20 patient cohort. And of great interest to us was that iron binding was increased in the bacterial populations in H. pylori infection compared to uninfected individuals. DNA methylation was also overrepresented as well. But again, this doesn't really tell you which bacterial species may be a driving force in pathogenesis. So we are fortunate because we had adjacent biopsies to the biopsies that were used for the microbiome analysis, and Dr. Fox's group has been a leader in cultivating low abundance bacteria from different GI tissues. And so they took the biopsies and they were able to grow staph epidermidis from the low risk biopsies that was not present in the high-risk group. They were also able to grow a strep salivarius species from the high risk, not the low risk. So the next step is to go into germ-free mice. So in our study that we published with them this year, these are germ-free mice that are infected with a strep salivarius from the high-risk region. Inflammation is minimally increased compared to uninfected controls. Dysplasia increased a little bit, not that much. As you would expect, infection with germ-free mice with H. pylori alone, increased inflammation, increased dysplasia. When you co-infect with H. pylori in this high-risk species, strep salivarius, you see a significant increase in the severity of inflammation and, importantly, the severity of dysplasia in these animals. Not shown on this graph is the fact that they also did co-infections with the low-risk bacterial species, staph epidermidis, and they saw less cytokine expression in the mucosa of these germ-free mice. So the last thing I just wanted to touch on was, we've talked a lot about the gastric microbiome. Is there a role for extragastric microbiome influences on H. pylori-induced host responses with carcinogenic potential? And again, Jim Fox's group years ago suggested that indeed intestinal bacteria can modify the severity of inflammatory responses in the stomach to H. pylori. In this study, they co-infected mice with intestinal Helicobacter H. muraderum, and then infected with H. pylori. They saw less inflammation in the stomach compared to H. pylori alone. Conversely, when they used another Helicobacter, H. hepaticus, they found more inflammation in the stomach. So it seems like intestinal bacteria may be able to modulate the severity of responses in the stomach. So I wanted to tell you a little bit about some recent work that we've been involved with at Vanderbilt. In my lab, Jenny Noto has led this effort. So we're really interested in dietary influences. Iron deficiency, because iron deficiency is a common problem worldwide, affects 30% of the population. H. pylori is linked to this, and iron deficiency is linked to premalignant lesions in the stomach. So what we wanted to do is try and understand some of the detrimental host responses that occur under iron deficiency with H. pylori. So we used the NSCAS mouse model, and we took mice, put them on an iron replete diet or an iron depleted diet, and then infected them with a strain of H. pylori that's CagA positive, PMSS1. And we found that in NSCAS mice that were iron deficient, you infect with H. pylori, you see significantly higher levels of dysplasia. So what's driving this from the host side? Well, we did metabolomics on these mice, and we found that there were several pathways that were overrepresented in the gastric mucosa of H. pylori-infected mice under iron deficiency. And one that we've sort of chased down in detail has focused on bile acid biosynthesis. So bile acids synthesized in the liver, but in the intestine, bacteria can convert these to secondary bile acids. And secondary bile acids have been linked to GI malignancies. Specifically, one secondary bile acid, deoxycholic acid. So we did some studies to try and directly look at the effects of DCA in this model. We took mice, made them iron deficient, infected with H. pylori, and we saw levels of this carcinogenic bile acid were only increased in this condition. It did not occur under iron normal conditions or uninfected mice that were iron deplete. We then directly gave the mice DCA in the drinking water. And when we did that and infected them with H. pylori, we saw a marked increase in the level of dysplasia. We can reverse this. As gastroenterologists, we use bile acid sequestrants very frequently to lower the bile acid burden in patients. So we did a study where we gave the mice cholestyramine to try and reduce their bile acid levels. We did that, and if you focused on the right panel, these are mice that are infected with H. pylori that are iron deficient. We see a lot of dysplasia. We see much less of that if we also give the mice cholestyramine. And then finally, one thing that we always try to do is move what we're finding in the lab into human populations. And so with Shalja Shah, she created a database for us, a retrospective database from VA patients, contained over 400,000 individuals. And our single question was whether treatment with bile acid sequestrants would alter the risk for gastric cancer. And indeed it did. So if these individuals use bile acid sequestrants for 1% of the days that we looked at, their risk for gastric cancer was reduced by 8%. If they used it for 5% of the evaluable days, the risk was 30% reduced. So we were able to validate in a human population what we had seen in our animal models. So why am I telling you this? Because intestinal bacteria are important in the generation of these carcinogenic secondary bile acids. So we also wanted to see in this study if iron deficiency could modify the composition of the microbiome within the context of H. pylori infection. So we took fecal samples from iron replete animals or iron depleted animals and then looked at their intestinal microbial composition. And we found that there was a clear separation in terms of beta diversity, community structure in the microbiota from animals that were iron replete in yellow or iron depleted in green. And when we started to look at some of the specific bacteria that were responsible for these changes, we found that there were only really 11 that were differentially abundant. These fell into the phyla of Firmicutes, Bacteroidetes, and also Verruca microbia. Why is this important? Because these bacteria have been reported to harbor bile acid inducible operons, bile salt hydrolases, that can convert primary bile acids to secondary bile acids. So we're now working with Jim Fox to try and grow some of these species up and sequence them. So I think that we have to move from what I used to consider the tragic triangulation of H. pylori-induced gastric cancer to a bad quadrangle that includes also effects of the microbiome. But we're really forced now as investigators to not only just categorize these elements, but also to understand the specific interactions that occur between them all so we can fully understand their effects on gastric cancer. So where do I think some of this work is going? I think future investigations will focus on defining microbial communities in well-characterized longitudinal human populations. A lot of the work that's already been done has been in cross-sectional studies. Now, if we're able to stratify individuals that progress to more severe lesions or remain stable, comparing those microbial compositions may be very, very informative. And one way that they'll be informative is it'll allow us to test high priority candidates in tractable models like germ-free NSCAS mice that recapitulate the human carcinogenic niche. But I also think there are clinical ramifications that come from this work. I think that these results will ultimately potentially allow us to identify persons who will benefit from H. pylori testing, eradication, and potential enrollment in a structured surveillance program, and also identify novel microbial targets for reversing progression. And, you know, if you think about it, you know, when we treat somebody for H. pylori, our endpoint is to confirm H. pylori cure. That may not be enough. We may at some point have to establish that that person, if they have atrophy, are able to have a restorative function of their GI mucosa. Because if it's the gastric microbiota, after you eliminate H. pylori, that continues to drive the later stages in this process, then we will not know that by just testing for H. pylori eradication per se. So I'll go ahead and end there. I'd like to thank all the people in my lab that have contributed to this work, our wonderful collaborators at Vanderbilt and beyond, and I've highlighted a lot of people who are here at this meeting who have helped me in ways I could never thank them enough for. Thank you so much.